Okay, hey guys, so welcome to the second half of chapter 1. In this video, we're going to look specifically at one type of structure within cells called the plasma membrane, how it works, and then how it functions within the overall purpose of the cell. Okay, we're going to pick up from where we looked at last time about cells, and then specifically information about how the function of the plasma membranes helps the cell survive. Okay, so let's jump into it. Chapter 1, the plasma membrane. So the plasma membrane. The plasma membrane also has a second name called the cell membrane, okay? And basically it is the boundary layer of the cell. It's like the skin of the cell, except it does a lot more than just separating the cell's internal environment from their surroundings. That's the primary function of it, but it regulates a lot of that movement, yeah? Now there are 4 characteristics you want to look at when it comes to the plasma membrane, and each one of these characteristics allows it to function the way that it does, okay? It's not just a wall, okay, or it's not just a skin, but it does things and it interacts with its environment and allows the cell to function, okay. The first is a particular term called semi-permeable or selectively permeable, yeah. What that means, it allows some substances to go in or out, but not others, yeah. Therefore, the plasma membrane regulates the entry and exits of materials in and out. When I say materials, I mean the small things that the cell needs to survive, and also the things it needs to get rid of, like nutrients, waste, chemicals, hormones, and etc., okay? It also retrieves materials and nutrients into the cell. Yeah, so it regulates things that go in and out. It'll pick up all the stuff that the cell needs and brings it into the cell, and then it removes all the waste out of the cell, yeah? The structure of the plasma membrane, the structure is what determines its function. In a lot of biological concepts, the structure helps us understand why it works the way that it works. Okay, so understanding the structure of an organelle like the plasma membrane will help you understand how it functions and how it does the things that it does, okay? So let's take a look, okay? The plasma membrane is made up of 2 major components. The first one is what we call the phospholipid bilayer. Now this word here, phospholipid refers to the structure, and the word bilayer meaning 2 layers, yeah? And the second thing is the proteins. Now, I would think of it as the phospholipid bilayer meaning the main boundary along the cell, and then the proteins are embedded along the bilayer. It's found within at certain spots over the boundary layer, okay? Here is a diagram of what the cell looks like and the plasma membrane structure, okay? The cytoplasm refers to the inside of the cell. All the components within the cell is called the cytoplasm. The word cyto refers to cell, and plasm just refers to like the plasma or the goo that's inside the cell. Yeah, so all of the stuff here is inside the cell. The extracellular, extra meaning outside, and cellular meaning outside the cell. So the external environment outside of the cell is here. Yeah, so if this was your cell or my cell, for example, if this was a blood cell or perhaps a tissue cell, there would be the internal environment of the cell, and this could be like the external environment like the arteries or whatever, or the fluid in the blood. And then this is the boundary layer, okay? You can see there all these little circles that form the phospholipid bilayer is in the creamish color, and then embedded in bits and pieces here and there are these proteins that have particular functions, each one slightly different from the others, but they're embedded like kind of spots, motted here and there along the boundary layer, okay? We're going to go through the structures of both, what they do and their particular purposes, and then we're going to find out how they work to allow the plasma membrane to regulate things that go in and out of the cell. Now this is picking up from what we looked at in the previous section, where we looked at how the size of the cell is really important to determine how things go in and out, and now we're looking at how the actual things go in and out of the cell, and why that's relevant, okay? Phospholipid bilayer. This is what it looks like, okay? The phospholipid bilayer is composed of 2 major molecules. You will find in biology that there's a lot of chemistry involved, and we kind of briefly skim over a lot of the chemistry, but it is still useful to understand it from a chemical level because it supplements your understanding of biology and biological concepts, okay? So here is the phospholipid molecule, okay? It is a chemical structure of a phospholipid molecule, phospho for the phosphate and lipid meaning a fat, or oils, yeah, so they form a major category called lipids. So here is the phosphate group, okay? The phosphate group forms what we call a polar head. Yeah, it is the head of one of the components, and then the lipid chain forms the tail. It forms this kind of two-pronged tail that sits at the bottom of the polar head, okay? This is the chemical structure, but the chemical structure then allows it to do something quite unique, okay? The polar head is attracted to other polar heads. So this circular thing at the top here, here's the actual chemical symbols that are behind it, but I'm not going to go into detail about that. That's just called the phosphate group. And this polar head is attracted to other polar heads. So in this diagram here, you've got one polar head that's attracted to the next one, right? The non-polar tail is also attracted to non-polar tail, see? So you got that one, that non-polar tail attracted to that one there, right? And so what you've got is you've got one side of it, one half of it, sticking the head sort of outwards from the external environment there, and then the tails are sticking inwards, and then on the other side you've got the kind of like an inverted structure where you've got the head sticking outwards again, and then the tail sticking on the inside. So now you've got a bunch of polar heads that are attracted to one another on the outside of the environment, and then a bunch of non-polar tails on the inside that is attracted to each other. Okay, now that structure is really important, so you got to keep mind of that. The structure allows this particular molecule, these molecules, to actually be really flexible at body temperature, yeah, which is about 36.7 degrees. The lipid chains are viscous fluid. This means that within your body, this plasma membrane is really fluid. Yeah? It tends to be very flexible and it can change shape. Yeah? And the reason for that is because this attraction force between the tails is not a fixed attraction force, not like, you know, when you look at, for example, in chemistry, you look at covalent bonding or ionic bonding. It's not as fixed as that, okay? It's just attracted to each other through a polar-polar attraction, and then what that does, it allows this bilayer to become really fluid at body temperature. In other words, you can bend it, you can shape it, and it can change according to the size of the cell, right? So there's actually nothing fixing them in place. However, what we generally find is that this particular polar head is what we call hydrophilic. In other words, it's attracted to water. Hydro meaning water, philic means to love. Yeah, so hydrophilic means to love water. Just like how, you know, if I was to say, you know, a pedophile is if someone a pedo refers to kids, phile is someone who loves kids. Yeah, I know it's a bit of a weird example, but that was the only one that I can come up with, right? The word hydrophobic meaning phobia or fear. So it fears water. So what you've got here is the polar heads are actually attracted to water, but the non-polar tails are not attracted to water. So what that actually means is that this little middle band here is going to prevent as much things that are polar from getting through it, like water, alcohol, and a whole bunch of other things as well. Okay, and so it's going to stop certain things from getting through, but it's going to not stop all things, right? Here is an example of what this viscous fluid looks like. Here's a capillary, yeah, within some tissue, and you can actually see there, these red blood cells don't stay in their fixed shape, yeah? The plasma membrane, the layer that surrounds them, can actually change shape according to the needs of the blood cells. So if this artery was going through a bit of a kind of a squeeze at certain sections, like for example along here, and possibly along here, and along there, right? The plasma membrane can then change its shape into a viscous shape, and then it can reform its original size according to the need. So it allows some measure of change. It's also what allows your body to move the way that it does, because you know, every single cell in your body is surrounded by this plasma membrane, and each cell can be bent and shaped and kind of, you know, compressed just a little bit, and that allows you to move your muscles the way it does. It allows you to move your body the way it does without kind of just breaking or falling apart in the cells, okay? So that's the phospholipid bilayer, and this forms the kind of the basis of all of your cells in your body, okay? It's also another reason why you need lipids or fats in your diet, because the fats that you take in your diet is also what forms the nutrients you need to actually form this bilayer, which is the non-polar lipid tail, okay? Now, the second thing in the bilayer is called the proteins. Now, proteins are divided into a whole bunch of different categories, but they're mainly categorized as being either integral or peripheral, okay? So the integral proteins are embedded in the bilayer, and they span the whole width of the plasma membrane, and we call that particular characteristic a transmembrane. Trans meaning to move across, membrane means to move across the entire width of the plasma membrane. And what they do is they play a really important role of what we call forming these channels and transporting materials in and out and creating receptors or signals from outside of the cell. Okay. The second component is what we call the peripheral proteins, which are anchored on the outside of the plasma membrane. So you've got these main channels which form your gateways in and out of the cell, and that's why they're integral. They're really important in allowing things actually going in and out through the integral proteins. And then the peripheral ones, not that they're not important, but what they do is they're not part of the plasma membrane structure, but rather they're attached to the membrane, and then it assists in moving things in and out of the integral protein. I'll show you a few examples and diagrams in a second, okay? So if you were to say, this is your phospholipid bilayer, see, there's the polar head, non-polar tail, facing each other like that, right? And then what you've got here is this integral protein that is like a gateway, right? So if something is too big to fit through the phospholipid bilayer here, then what it has to do is it has to go through this particular channel to get through. And to get through the channel, it needs to go through some sort of chemical reaction, which we'll explain how it does that later on in this PowerPoint. And then it is assisted by what we call a peripheral protein that allows that process to happen a bit more smoothly. Yeah, they're like your little lackeys who move in and out and assist in the chemicals to move in and out of your cell, right? Now, together, the proteins and the phospholipid bilayer forms what we call the fluid mosaic model. The fluid mosaic model is the name of the model they give the plasma membrane, yeah? And so it suggests that the plasma membrane is two-dimensional. It's this flat layer, and the proteins are embedded. Basically, everything that I've told to you guys about the plasma membrane up until now is part of the fluid mosaic model, okay? The reason why it's called a fluid mosaic model is because it's fluid. The lipid chains within the bilayer allows some sort of molding and change in the shape of the cell. And it's mosaic because the mosaic refers to those proteins forming that mosaic pattern that's mottled all over the plasma membrane, okay? And so that's the same diagram that I've been showing you before. Once again, here's an integral protein. Here's a different type of integral protein for a different chemical. And then here are the peripheral proteins, yeah? So a good example would be, if, for example, you drank a lot of water. Now, water is so small that it can freely move in and out of your plasma membrane a bit easier, which is why your cells tend to swell up a bit more when you drink a lot of water or when you are surrounded by a lot of water and you get like wrinkly skin and things like that. However, if you were, for example, to try and take sugar and move it from outside of the cell, inside of the cell, the sugar cannot go through this particular layer, and so it has to go through the integral protein there, right? If you, for example, ate meat and your body digested the meat and turned it into protein, the protein themselves can't go through the plasma membrane either. So they have to go through another set of integral protein. And so the sugars will go through one channel and the proteins will go through a different channel again, okay? And each one having their own set of peripheral proteins to assist that movement in and out of the cell, okay? And so you can see there, your plasma membrane needs to have channels for all different types of material that is moving in and out, and so it's a very complicated process, all right? The functions of the plasma membrane, we've already talked about how it functions. I'm going to kind of formalize the functions. The first one is called what we call the active or selective boundary, okay? It separates the cell from the external environment and it only allows certain materials through. That is an active thing. It uses energy to move things through. That's why it's active and selective, because it uses energy to allow some of those things through. The cell also is responsible for identifying the cell as part of your body, right? So it's responsible for cell identity. Particular molecules called glycoproteins, glyco for sugars, and proteins for proteins. And so a glycoprotein is a combination of a sugar and a protein put together. And what they do is they act as surface markers for the cell. So it acts as a marker and it allows the immune system to identify self from foreign cells. That's why your immune system is not going to attack its own blood cells, but it will, however, attack a pollen or something like that that's made itself into the lungs and so forth, okay? These markers are called antigens, and we'll talk more about antigens in the later chapters, and particularly also in Unit 2, right? The plasma membrane also receives signals. Yeah, those proteins can act as receptors. The transmembrane proteins will act as receptors for signals. And when the signals attach to the protein, it changes the shape of the receptor protein, and then it changes the way the cell functions. That's what hormones do. Hormones change, elicit some sort of response in the cell by attaching themselves to these proteins and causes the cell to change its function in some sort of way. That's why your body changes the way that it does when you go through puberty and you have lots of hormones like testosterone or estrogen, okay? And the last one is that it transports materials. Yeah, that semi-permeable allows it to move some substances across. Some substances can always, others can only move across with the assistance of those proteins, okay? We're going to mainly focus on how things move in and out of the plasma membrane. So methods of crossing the plasma membrane. There's a whole variety of things. If you think about the stuff you eat in your diet, and you think about the kind of molecules that your body needs, right? It can span anything from proteins to sugars to fats to, you know, vitamins and minerals. Your body needs to regulate all of those things. However, it can't just let everything through all the time. It has to manage what's going to go through and when's it going to go through and how much of it is going to go through, because your body then uses those proportions for whatever function that it needs to use it for, okay? And so how easily the molecules can cross the plasma membrane depends on a few different factors, okay? This table is taken directly out of your textbook. Very useful kind of oversight, and I just need you to understand the different factors that play a really important role in determining what molecules will cross and which ones can't, okay? The first and most obvious one is the size. Smaller molecules tend to cross easily compared to larger molecules. So and when I say small and large, they're still very, very, like microscopic in terms of their size. You can't really look at them with the naked eye. However, we divide between micromolecules and macromolecules, okay? So macromolecules are things like proteins. They're huge, yeah? Nucleic acids, which is what makes up your DNA, also are too big to go across the plasma membrane. However, smaller molecules such as gases and sugars and water will cross a little bit easier. Not to say that they're all going to cross easily. The second thing is what we call net charge. Yeah. Now remember when I was talking about the phospholipid bilayer, I was talking about how the polar head is attracted to other polar head. If something is polar, it means it has a charge. It's either a positive charge or a negative charge. If it's non-polar, it contains no charge. And so if something has a net charge, either a positive net charge or a negative net charge, what that also means is that it makes it more difficult to cross the plasma membrane. If it is an uncharged molecule, it tends to cross it a little bit easier. Or if it's a gas, for example, carbon dioxide and oxygen will cross the plasma membrane more readily. If they didn't, you would have a lot of trouble getting oxygen and carbon dioxide in and out of your lungs into your cells, because it needs to cross the membrane in your lungs in order to do that, right? And so gases will cross a little bit easier. Uncharged molecules, such as urea, which is what your urine is made up of, ethanol, which is the compound in alcohol, right, can cross the plasma membrane a little bit easier. That's also the same reason why you get drunk so easily, right? Because ethanol is so small, it'll go straight through, your stomach lining and straight into your blood, and then it carries around straight into your cells, and then it gets you drunk, okay? In contrast, minerals such as potassium, sodium, and chlorine can't cross as easily because they get repelled by that hydrophobic lipid, or that non-polar lipid tail of the plasma membrane, okay? The next one, solubility. How soluble something is in lipids or in fats will also then determine whether or not it can move across. Yeah, glucose, for example, cannot cross because it gets repelled by the hydrophobic lipid component. It's small, but what it is it's not soluble in lipids, and so it doesn't cross that boundary because the lipid bilayer stops it from going through. It has to go through the protein channels, okay? And then also how much of it the cell already has, what we call the concentration gradient, yeah? Generally, if there is more outside of the cell than inside of the cell, things will move into the cell without needing a lot of energy. This is not every one of the cases, but this is a general trend. And then also if there's more inside the cell than out, things generally move out of the cell, okay? And I'll go through that later on as well. Now, what's the problem with some of this? When you look at some of these different molecules and some of the descriptors, how do we know and determine these things, and what's some of the problems that we see there? One of the key issues is that for some molecules like glucose, sodium, and calcium, they're all essential and really important, but they can't cross the plasma membrane on their own. So the cell needs to actively take them in. That's why we call it an active or selective boundary, not some sort of passive one, because it is actively picking up certain molecules and rejecting certain others, okay? So that the cell can function the way that it needs to, right? And so it just means that there needs to be another way through the protein, okay? Here's a diagram overview. Gases, very readily. Small uncharged molecules like alcohol or ethanol will move through very readily. Other things such as urea and water sometimes will go through, sometimes won't. Glucose is too big. Ionic compounds, things that are polar, will also be removed, and charged molecules that are large, macromolecules like protein, amino acids, and things like that are also too big, okay? So that's just a bit of a general overview. We're going to look at what happens when it's too big. Yeah, when it's too big and it can't fit through, how is the cell going to retrieve some of those things that it needs? Okay, and that's what this is looking at here. Small things we'll go through by a method of what we call simple diffusion. Simple diffusion is the same as diffusion, but just in cells. Yeah, diffusion is defined by the movement of particles from a high concentration to a low concentration. If there is lots of water inside the cell, then outside of the cell, water will tend to move out of the cell. If there's lots of water outside of the cell and less inside of the cell, water will tend to move inside the cell. Okay. The next step is what we call facilitated diffusion, right? Facilitated diffusion is where you still have movement of particles from high concentration to low concentration, but it has to go through the protein gateways. It has to go through the integral proteins, yeah, the transmembrane proteins. It still goes from high concentration to low, but rather than moving straight through the plasma membrane phospholipid bilayer, it goes through the protein channels, okay? Now, just before we get on to active transport, simple diffusion and facilitated diffusion, all methods of diffusion doesn't take energy from the cell, yeah, because it's moving from high concentration to low concentration. It is a natural process. It naturally happens, and so what happens is the cell will naturally absorb things either into the cell or out of the cell because of the differences in their concentration levels between them, and that doesn't take any energy on the cell's behalf. If the molecules need to go up a concentration gradient. That is, it needs to go from low concentration to high concentration. In other words, the cell wants to take as much of it as it possibly can. Then what it needs to do is a method called active transport. Active transport is using energy to actively move molecules through the protein gateways or the integral proteins, and it takes it into the cell or it pushes it out of the cell. Okay. Now, these particular protein transporters are known as pumps, and what they do is they move it up the concentration gradient. They're moving it against the amount that's there already, and so that takes a lot of energy. Right. The last one is what we call bulk transport. Bulk transport is when cells want to move very, very large macromolecules and liquids in and out of the cell, and so they actually create a small package that gets delivered into or out of the cell. Now, if it's going into the cell, it's called endocytosis. Endo meaning inwards, cytosis meaning into the cell, right? So, and exocytosis meaning out, and cytosis of the cell. Exocytosis moves out of the cell. I'm going to go through these in more detail, but this is just a bit of a summary, okay? Already said that. And that's a diagram of what endo and exocytosis looks like, yeah? Quite literally, the cell pinches a small little packet called a vesicle from the plasma membrane, and then moves it into the cell like that. And then when it needs to throw stuff out of the cell, it merges into it and it releases these molecules out of the cell. Okay. Now let's go through simple diffusion. Defined as the movement of substances from an area of high concentration to low concentration down the concentration gradient. That down the concentration gradient is important to realize. Okay. Simple diffusion is the same process but in cells. Okay. It requires no energy. It's completely random, but it eventually makes its way down. Yeah. So if, for example, you started with a whole bunch of molecules, for example, water molecules outside of the cell versus inside, due to the random movement of the molecules alone, over time, you would begin to see the balance between inside and outside even out. Okay? This mid-band is called the semi-permeable layer or the plasma membrane. And so naturally, what's going to happen is eventually you're going to get to a place where both the concentration inside of the cell is the same as that of outside of the cell, right? And so that's diffusion at work. You leave it long enough and it will balance out on both sides, okay? Diffusion happens everywhere, okay? But when it happens with water, which is, a really important substance in living things, we call that process osmosis, yeah? Osmosis is not defined the same way as diffusion, but the process happens in a very similar manner, okay? Osmosis is defined as what we call the net movement of a solvent, which is usually water, across a semi-permeable membrane from lesser solute concentration to higher solute concentration. Now that sounds really confusing, but it is really just defined as the diffusion of water. When water moves from one area of high concentration to low concentration, it is osmosis, okay? However, the way that water moves is a little bit different because it is defined by solutes, yeah, and ions and stuff like that, okay? Now, just a bit of a recap on Year 8 science for you. A solute is the substance that's being dissolved, and then a solvent is the liquid that it's being dissolved in, okay? So the solvent in this case is water, and solutes could be things like salt or water, sugar and things like that, okay? The solution is when you mix the two together, okay? So what happens is, if I was to show you osmosis happening, right? Let's imagine you've got a beaker and you put in between the beaker a semi-permeable membrane. Now this could be a piece of, a plasma membranes of a cell, a piece of cellulose paper or something like that. And what that is means that this divide is big enough or has big enough holes that allows water to go through, but it doesn't allow this big purple molecule, which in this case, let's call it sugar, right, to go through. So if you even the beaker out, you put this right in the middle, and on the left side is very, very few sugar molecules dissolved within the water. But on the right side, you've got a very concentrated amount of sugar molecules in the water, right? And you want to track how the water moves, right? So what's going to happen over time is that the water is going to move from an area of lesser solute concentration, that is lesser sugar concentration, to higher sugar concentration or solute concentration. So if the water started out evenly on both sides, it's going to move from where there is lesser solute concentration on the left here to the right side, which is where there's more solute concentration, because the water is attracted to the sugar molecules. It's going to move through this red line and moving to the right, and what you're going to get is it's going to balance out the concentration levels of water compared to sugar, right? So, you've got 1, 2, 3, 4, 5, 6, 7 molecules on this side, and, probably about 30 or so molecules on that side. The water's not going to stay evened out. It's actually going to move to the right, and it's going to fill the right side to balance this out. Now, the reason for that is because the sugar molecules can't cross this red line, because it's semi-permeable, and so they stay on one side, and the water is actually attracted to the opposite side to all of those sugar molecule concentrations. You can do this experiment at home if you have a piece of cellulose paper that you can just buy from Coles or something like that, and you pour one side with sugar, dissolve it, the other side with less sugar, and then dissolve it. The water's going to move to wherever the high concentration of sugar or salt or whatever is, yeah? That's osmosis at work, and osmosis plays a really important role in cells, because osmosis is the reason why your cells will dry out or will swell and things like that when in different concentrations of salt and water. Okay, depending on the condition, we have different terms for it. Okay, you might have heard some of these terms probably in a more sporting context, but biologically they all mean different things. Okay, so the word isotonic in osmosis refers to when the solute concentration is the same as the outside. Whatever you've got inside of the cell is the same as outside of the cell, and so it is balanced. Iso meaning the same, yeah? If you ever go kind of like reading weather and stuff like that, you hear the word isobars meaning the same pressure. Isotonic, tonicity refers to the concentration of solute. So isotonic means it's got the same concentration of solutes in and outside of the cell. In other words, the water moving into the cell is equal to the water moving out of the cell, and therefore it's balanced. There's no movement or net movement of water, and the cell neither swells nor shrivels up, okay? So this is if the outside of the cell is isotonic, then water in is equal to water out. However, if the solution is called hypertonic, hyper meaning high, tonic meaning solute. So if the concentration is greater outside than inside, then that cell is in what we call a hypertonic solution. And what that actually means is that water will move out of the cell, right? Because there is more like sugar or salt outside of the cell. The water is going to trail and try to follow that amount, and it's going to result in a net movement of water out of the cell, causing the cell to shrivel, okay? Hypotonic is when the concentration or the solute concentration is lesser outside than inside. In other words, you're in an environment where there is like very, very little salt or sugar or solute outside of the cell than inside of the cell. And so water is going to move into the cell, causing it to swell and eventually burst, okay? Here's a diagram to show you what I mean, okay? Let's say you take a red blood cell, you isolate it, and you throw it in a beaker full of salty water, all right? The red blood cell already has a certain concentration of salt in it. If that concentration matches the outside environment, we say that the red blood cell is in an isotonic environment, and the amount of salt that it's in this case here, is the same outside of the cell and inside of the cell. That means that the same amount of water going into the cell will match the amount leaving the cell, and the cell is in a stable environment, okay? If you put the red blood cell in a concentration of fresh water, like pure fresh water, and you take out all of the salt, with the exception of a few, the red blood cell is now in a hypotonic, meaning low tonicity, a hypotonic environment. And what that means is there's more water concentrated out of the cell than inside of the cell. And so water's going to move into the cell to try and match the salt concentration in the red blood cell, causing it to swell up in water. And eventually it's going to burst, right? And so it's going to burst and it's going to break, right? And so that cell is now in a hypotonic environment, okay? If we took it and put it in concentrated sea or salty water, then you're going to get the opposite. You're going to get a cell that is in a hypertonic environment. And what that means is that hypertonic, there's more salt outside, and the water is going to leave the cell at a faster rate than entering the cell, and therefore causing it to shrivel up and die. Now, this is why you kind of see those ads for Gatorade and Powerade talking about isotonic drinks, because what happens is when your body goes through rigorous exercise, you sweat, and when you sweat, you lose salt from your blood, and so your blood is now becoming a hypotonic environment. It's a hypotonic environment because your red blood cells are now in blood and your blood, and it doesn't have enough salt in your blood or your cells, and it's in a hypotonic environment. And so what's going to happen is it's going to lose the water concentration that it's in if you are not being careful. So what generally happens is you then need to drink some water, but you need to drink water with salt in it to replenish those lost salt that you've got from sweating. And so that's why they advertise for isotonic drinks, because the isotonic component of it is full of kind of salt in it that allows you to replenish some of the ones that you have lost when you sweated, okay? They also put a stack of sugar in there, so it's not really good for you either. So your best bet's actually really just to take a pinch of salt, chuck it in some water and drink it. Not concentrated amounts, but just a small amount, right? Alternatively, before they had fridges, what they used to do is they used to chuck food like meat and things like that in a lot of salt. And what would happen is if you put it in just salt, no liquids, pure salt, the amount of concentrated salt that's there is an extremely hypertonic environment. And so what it does is that, if you chuck your meat in this, all the water is going to leave the meat and it dries up, right? And that's why you kind of get dried meat when you salt a lot of your meat. But the good thing about that is when it dries up, there are other things like bacteria that will also be exposed to this hypertonic environment, and they will dry up as well. So the bacteria can't actually get to the meat break down and make the meat rot because they get dried up by the salt and they die before they can affect or eat through the meat. And so you've got this dried up, well preserved piece of meat because whatever bacteria is in that environment is in such a hypertonic environment that it dies before it can actually get to the food, right? And so that's kind of a bit of a contrast between your two different environments and understanding the role of osmosis there. Now, for the body, being isotonic is really important. You don't want to be in a hypotonic because your cells are going to swell up, and you don't want to be in a hypertonic because they're going to shrivel up. So you can only remain in these states for temporary periods of time, but for the most part, it should be water in equals to water out, and therefore you must be in a stable isotonic environment, and your body works really hard to make sure that happens. If you have too much water, your kidneys remove some of that water, and then you dump it out, and you pee white. If your body has too much salt or like caffeine or sugar or whatever, your kidneys then reabsorb as much water as it can, dumps out the salt and the sugar, and your pee turns out yellow, right? And so that's how your body maintains its tonicity within its blood and its cells. Okay. All right. Facilitated diffusion. Facilitated diffusion also doesn't require energy from the cell. However, it's mainly used for molecules that are too big or charged to go through the phospholipid bilayer on their own. So they need to go through a channel protein because the channel protein or the integral protein is large enough to fit the molecule, but it's not too large and it doesn't require energy. It just freely moves in and out, right? Here's what it looks like. Yeah, so facilitated diffusion. In simple diffusion, they just move in and out. In facilitated diffusion, they move in and out freely, but they do that through a channel protein that spans the transmembrane protein, and it moves it in and out like that. Okay. Now, there's a second set as well of proteins called carrier proteins. Now, these carrier proteins also allow things to diffuse in and out, but they're kind of like this special switch that switches on and off when the molecule is attached. This restricts the amount that can go in and out. Very few can go in and out like that. But what happens is it goes in, and when the molecule goes in, it creates a chemical reaction inside of the protein that triggers a release on the other end, and then it gets released inside of the cell or vice versa outside of the cell, depending on what the cell's doing. Okay, so these carrier proteins are a little bit different. This process is slower, but it's more restricted. It's not allowing just anything to squeeze in and out, but it restricts that movement. Yeah. Now, in real life, these molecules are, big chemical molecules, but here you've just got a nice little diagram that shows you what it looks like. Okay. This little pore here is filled with water. And so what they do is if they're charged or too big, even though the phospholipid bilayer is uncharged in the middle, this water space here and this water space here is charged. So it allows things like sugar to dissolve within this pore and move through the cell naturally. Okay, so it doesn't require energy, but it is slower because if you think about it, the amount of plasma membrane phospholipid bilayer available is so vast that small uncharged molecules can just go straight through simple diffusion. But then, the size of these molecules, there's only so many channel proteins that the molecules can go through. And so diffusion versus facilitated diffusion look a little bit different, right? I've mentioned this already, yeah? So the channel proteins are transmembrane proteins that have a central water-filled pore, and it allows charged molecules to go through. They don't normally cross the phospholipid bilayer because they're either charged or polar, so this channel allows it to bypass the bilayer and diffuse into the cell. The carrier proteins allow specific molecules, usually uncharged large substances such as glucose and amino acids and ATP. You will learn what these things are as we go along, and each carrier protein in the plasma membrane is designed for one type of molecule they want to transport. In other words, you've got a certain set of carrier proteins for sugars, a certain set for amino acids, and a certain set for ATP. Okay. When the molecules bind to the carrier protein, it changes the shape, opens one end, closes the other end. That's the way it's regulating the movement into and out of the cell. Okay. If we compare the rates of diffusion, simple, usually takes a bit of time to get started, but you can sort of see here, if I was to show the rate of diffusion, it's linear for simple. Yeah, naturally they will move over time. The concentration gradient will even it out, and they move through naturally. Facilitated, on the other hand, starts off a little bit faster, but it slows down because once it gets crowded and those channel proteins of peripheral proteins need to sort of end up being clogged, and so they limit carrier proteins, channel and carrier proteins, they become limited, and so they get full. Once it becomes full, the diffusion then slows down quite dramatically, and so you can see it tapers off at a certain point, right? Which is why your body can only pick up so much glucose or so much of a particular type of uncharged molecule at one time through facilitated diffusion, right? It's too slow, right? And that brings us to the next part, which is active transport, okay? Active transport is moving substances across the plasma membrane against their concentration gradient, from low concentration to high concentration. Now, this particular process requires energy. Yeah, that's why it's called active transport and not passive transport. The first two that we looked at, diffusion and by extension osmosis and facilitated diffusion, are passive transport methods and require no energy. But in this particular case here, active transport is an active process and requires energy. And it requires energy in the form of a molecule called ATP, adenosine triphosphate. Okay, ATP is the kind of the currency for cells, and it is the energy package that cells use as a bit of a monetary currency to function the way that it does. Okay, one type of active transport channel is what we call the sodium-potassium pumps. Okay, these sodium-potassium pumps are really important for nerve and muscle function. Okay, when your brain sends out a nerve impulse, it releases a polar molecule called sodium, and the channels need to open and close, and what happens is the sodium floods into the cell and potassium floods out of the cell, and it allows the message to pass on from one cell to another, from one nerve cell to another nerve cell, and that's what allows your body to send signals around. Yeah, now this happens really fast because you think about it, you know, the moment you go, I'm going to lift my hands with your mind, not that you ever think I'm going to lift my hands, your hand automatically lifts, right? And so there needs to be a signal sent from your brain to your hand muscle, the muscles in your hand, that allows it to lift straight away without really, you know, it's almost like a subconscious kind of thing. That's how fast the nerve impulse actually is, okay? Now, active transport is really useful on a few different occasions, okay? And I'm going to go through a few examples of why it's useful and explain why your body needs to not only allow things to move in and out passively, but actively take up some things compared to other things, okay? A good example is glucose. So glucose is a sugar, and it is the most important one because glucose is where you get your ATP or your energy from. You get your energy from eating sugars. That's why little kids go on a little bit of an energy high when they have too much sugar before, you know, at nighttime, okay? When you eat a meal, say for example, you grab an ice cream from the ice cream store and you eat the ice cream, it gets into your stomach, it breaks down in your stomach, gets to your small intestine, and it starts to absorb the glucose from those sugars that you've just eaten, right? What's going to happen is the intestines are on one side on the left here, right? Your blood is on the other side, and that's what allows it to transport the glucose around your body. But in between are the cells in your stomach lining. They're the actual cells that get exposed to your food and your blood at the same time. And so it moves stuff from your intestine through the cell and into the blood, okay? And so there you've got crossing the plasma membrane actually twice, yeah? Once into the intestine cells and once out of the intestine and into the blood, okay? Now, imagine if you had glucose in your intestines. What's going to happen is, if the process was passive and it didn't take any energy, naturally the glucose will still move into the cell through your transport proteins, yeah? It's going to move through these protein channels, right? However, right, if it was, for example, facilitated diffusion, it's going to eventually even out between the two processes, yeah? If, for example, the concentration here is matched with the concentration inside of your cell, if it was a passive process, what's going to happen is the glucose is going to stop moving in and out of the cell, because what's outside of the cell, the concentration, is the same as the concentration of glucose inside of the cell, right? Your body doesn't want that to happen, because when you eat food, right, you want to take as much glucose out of as possible and leave none to be kind of pooed out the other end, right? You want to take as much sugar as possible from your diet or as much proteins as possible from your diet. And so your body needs to keep actively taking up the glucose, everything out of your intestines and into your cells, and then push all of that in the opposite direction out into the blood as possible, so that there's no balance between them, right? And so that kind of up the concentration gradient is an active process, okay? That is an example of active transport. There's a few other examples. So for example, in plant roots, plants want to take up a lot of minerals. Yeah, they want to take up a lot of minerals because the minerals is what allows them to grow, and they need the minerals for certain processes within their metabolism. However, if it was through passive transport, once again, the minerals in the soil matches that within the plant root cells, the plant's going to stop taking up minerals, and it doesn't want to do that. It wants to keep taking up minerals, and so it needs to actively suck up the minerals along with the water in the soil around it, and as a result of that, it's going to use active transport. Okay. The third example is the acidity of gastric juice. Gastric juice is the same acid that is in your stomach that allows your stomach to break down things the way that it does. Yeah, gastric juice is made up of a particular molecule called hydrochloric acid, and your body needs a high concentration of this gastric acid or hydrochloric acid, and to do that, it needs to keep pumping the acidity of your stomach up and up and up, so that your stomach becomes more acidic as it goes. If it stops that and it was an active process, then it's going to balance out, and the pH inside your stomach, in your stomach versus the pH inside your cells is going to match and sit at the same level. But because your body needs to keep pumping out the hydrochloric acid, it brings the pH down to like a 2 or a 1, making it more acidic, and as a result, the acidity of your gastric juice increases through the process of active transport. So your body is actually putting energy in to make your stomach that acidic, okay? Oops, this kind of just got broken off a little bit, but obviously the difference between the sodium and potassium pumps vary between them. Yeah, potassium sodium pumps are one of the many different pumps that you've got in your body that allows you to push that. Other things are for glucose, fats, lipids, proteins, etc., etc. Okay. Now, the last method, and the end of this video approaching, is looking at a method called the bulk transport method, or endo and exocytosis. Okay, bulk transport is when you move things in bulk. Yeah, solid particles and liquid particles need to be moved in bulk, in or out of the cell, are moved through a process of bulk transport. And what it does is a part of the plasma membrane actually pinches off and surrounds the material, forming a vesicle or a package. Yeah. This movement requires energy, but it allows the cell to collect large amounts of the material. Okay, bulk transport comes in two parts. When it's going into the cell, delivery into the cell is endocytosis, and that's what it looks like. Yeah, there's your phospholipid bilayer, and what happens is it forms a little fold, and then it sucks in that material. If, for example, if it was a large protein, for example, it sucks in heaps of segments of a large protein. And then what it does, it actually pinches off right at this point. And remember, the structure of the plasma membrane is very fluid. It allows changes to occur in the plasma membrane without destroying or breaking the cell. And so that little pinch that it performs right there forms a vesicle, and then the remaining parts actually reattach to itself, reforming the plasma membrane outer layer. And so that actually allows the cell to create a kind of makeshift package that contains the bulk transport materials that it's moving into the cell. Exocytosis moves things out of the cell, and so the opposite process, if it's a waste product within the cell that it's accumulated and it needs to get out of the cell, it will do a bulk transport of materials. If it's for example, the body has produced a certain protein within that cell and needs to be delivered to another part of the body, then it will shift that into a vesicle, move that through the cell, pinch to the plasma membrane, and then creates a fold and releases the particles outside of the cell into the blood and other tissue areas, and then moves it around the body as needed. Okay, so that's bulk transport, and we've looked at all of the different ways. Just to recap the different methods of crossing the plasma membrane, you've got diffusion or simple diffusion, and by extension you've got osmosis, which is the movement of water, and then you've got facilitated diffusion, and those three methods are what we call passive transport. And then the last two, active transport, is an active process where you use pumps or channel proteins to move things in and out. And then the last one is bulk transport, where a part of the plasma membrane pinches off, either moving it into the cell, forming or called endocytosis, or moving out of the cell, called exocytosis. Okay, so that's all of chapter 1. I hope you guys learned something new there. And in the next chapter, we're going to take a look closely at the different structures within the cell and how that works. Okay, I'll catch you guys later.